Good. All right, I think we are live. Reminders. Lab practical today. Check your times. Come to your assigned time. We'll have about 50 minutes for the practical. We'll try to start as close to 2 o'clock as possible. There's a total of 64 questions, so 60 points, plus there's 4 extra credit questions on there, so it's out of 60 points. Once you come in, you take it, you're done. So that's all we're doing today. All right, the lab's open. I'll double check on my way back down to make sure it's still open, but you have until about 12:30, 12:45 to review. At that time, I'll go in and I'll close the doors and I'll start getting it set up. All right. So lab practical. The exams. Target is to try to get those back to you within a week. It's always my target. So if I can get them back on Wednesday, I will. If it waits until Friday, then you'll get it back on Friday. She'll get it back this week. Any questions? All right. So we talked about this, right? Kind of unique structure, how the nematode muscles are attached. U-shaped projection on the cell body itself. All right, and then it connects to the striated muscle. So this part is not striated at all. So this part up here. All right, so I think did we talk about this? Maybe? No. Okay, so what's the function? One of the big ones is protection from the environment. And that includes protection from the host immune system, because the environment is just the immediate area where the worms are residing. So it could be what we see as the external environment, but it could also be inside the host body. All right, so one of the big things with this protection is that it is pretty impervious. It's hard to get things through that cuticle because of its structure, because it has the lipid layer, because it has protein surface, because you have this cortical zone. It's pretty tough. So it's going to be resistant to desiccation. But not only that, but the cuticle also provides protection against digestion. So as you'll see, some of our life cycles require the host to ingest these nematode larval stages, and when they ingest it, that nematode has to survive moving through the stomach, and the cuticle provides some of this protection. It's also the first line of defense against other pathogens and the host immune system. So we do know we have some viruses and bacteria that infect the worms. We know that cuticle is going to protect them in a way, and cuticle also protects against the host immune system. So that thin protein layer may act as kind of an immune system inhibitor, or it could capture the antibodies, which are then kind of sloughed off, much like the syncytial tegument would slough off. But again, we're not talking about the entire tegument. It's just a very thin protein-rich layer. So protection's a big part of the cuticle. The other big part of the cuticle is that it's important for locomotion. It's providing a rigid structure against which the muscles can actually work. And muscles aren't going to do work if they're not attached to anything. All right, so our muscles attach to the cuticle. So when they contract, you're actually pulling the cuticle in. But not only that, the cuticle's providing a good seal for the hydrostatic skeleton. So it's going to lend support to it. All right, so it's structural support. So it's important for locomotion and for protection. Those are, major ones. Now, the thing with this cuticle is that it's molted. as these worms grow, it's not going to expand and stretch. It's too tough for that. So it's going to have to break, and the worm's going to have to secrete a new one as it enlarges. And that is why nematodes are in the superphylum Ecdysozoa. What does ecdysis mean?
The outside dies.
The outside dies? It's a good guess. Anybody?
You digest outside of the body.
Good guess. No? No? Didn't you have entomology?
Yeah. No.
Who else had entomology? Did anyone else have entomology? Insects. Ecdysis is the term for molting. So yeah, this superphylum Ecdysozoa, it has all the organisms that molt, and the nematodes are in there because their cuticle has to break apart the worm, and then they have to resecrete it in order for the worm to grow. All right, and last part of this cuticle is that it's highly conserved. The cuticle, the shape, structure, basically conserved across the nematodes. So once you, in a way, once you've seen one cuticle, you've seen them all. All right, ready? Digestive system. So now we have a parasitic group that has a complete digestive system. Complete digestive system. What does it mean? It means you have a mouth and an anus. What's the benefit of that? What's that? All right, that's similar to platyhelminths. Get rid of waste. It's what? More space to get nutrients, and because you have that more space elongated, you can actually subdivide it to do specialized functions. So you can have maceration, right, breaking up of the food particles in one part of your gut, then you can have absorption in another part of the gut, and so forth. So it's really, having this complete digestive system is pretty good for acquiring nutrients. All right, we're going to break it down into different parts of the gut. So the foregut is that anterior part. This is the part that is cuticularized, so it has a cuticle. What's the consequence of that? So it's not really going to absorb food in that area, and when the worm grows and sheds its cuticle, foregut gets shed as well. So it's going to be shed and it's going to be regrown. So what makes up the foregut? Well, the mouth, the buccal capsule, and the pharynx. So the mouth is also called the stomodeum. It's kind of you could say your highly scientific term. I don't know if you would ever encounter that or not. Maybe in the MFAT, who knows? That's the mouth. Right? The buccal capsule is basically your opening region of that mouth. It could be simple, much like this worm is, where it's just a very small opening, or it could be complex, like composed of like cutting structures that we'll see in the hookworms. And then it's also the pharynx, which we're going to say the pharynx and the esophagus are kind of together just because of their structure. So when we look at it, we can break this down, and we will, into different parts of the pharynx and the esophagus, but usually we talk about esophagus, esophageal bulbs, and so forth as a muscular structure that's sucking in food and starting to force it down the gut. All right, so yeah, function of the pharynx, esophagus, suck food into the worm and propel it into and along the gut. So we're going to get movement, pushing action, because our intestine is not going to be doing peristalsis. All right, so that's our foregut. The midgut, did you guys get this? Oops, before we get to that, pharynx patterns, they're going to vary between the groups. All of them are basically triradiate. What does that mean? Yeah, basically, so if we were to look at a cross section, it looks like it has three-way symmetry, radial symmetry. And that's really a distinguishing feature when we do cross sections to say, oh yeah, that's the pharynx. And I don't think we have any like different cross sections where we're at the pharynx or esophagus. But it's triradiate. The esophagus could possess numerous glands. Could also possess an esophageal or a pharyngeal bulb. All right, so these are a couple different groups: Ascaritida, Strongylida, Oxyurida, Spiroritida. Right, you can have this bulb, esophageal bulb at the posterior end. Doesn't have to, because you can see the other ones aren't. You have some that have a slight narrowing and then a slight and then an enlargement. You have some that are fairly nondescript. With a ventriculus, almost like the stomach, and appendices. All right, and then you've got some glandular cells on some of them. It's all a variation. So just know that we could have a lot of variation in the esophagus type. The esophagus type actually helps us get to our IDs. All right, now we're on to the midgut. Midgut's uncuticularized. So this is going to persist with our worm as they grow. It consists basically of a single layer of columnar cells. Simple, single layer of columnar cells. And that means there's no muscles tied to it. And that's, you know, I put this here because if we look at human anatomy, you can see you have these cells. You've got these columnar cells. And then underneath them, you have your muscles because you have peristaltic action that moves the food through the gut. Not so with the nematodes. There's no muscles, so that means all of the food that gets moved through the gut is done so by the muscular esophagus, muscular pharynx. Not only pulling in food, it's also pushing it and forcing it through the digestive tract. When we look at this in cross-section, it's going to appear flat. So we said we have this triradiate symmetry with the pharynx. When we look at the intestine, you'll see it as kind of a flat structure like that. Usually. Sometimes it looks more circular. Again, just kind of depends on how the worm is fixed or how it was mounted on the slide. That's a midgut. It's going to likely run most of the length of the worm. All right, the hindgut is cuticularized. Again, consequences, well, means we're going to be shedding it. When the worm molts, we also shed it. Now, the ending of this worm is called the subterminal proctideum. Really highly scientific term. All right. So subterminal, what does that mean? Yeah, we're not at the end. We're subterminal. So we're going to find it. All right. What this proctideum means, or what it would refer to, that would be the anus in the females. That would be the cloaca in the males. That's our termination point, but we do utilize some different terms because this opening is the urogenital opening for the males. It's both the digestive tract opening and reproductive system opening for the males. For the females, it's just the anus because females will have a vulva that opens into their reproductive system. All right, so that's kind of a big distinction. And you do have muscles. You've got this rectal dilator muscle. I don't even know if I'm ever going to ask that. But point is that you've got a muscle that keeps it closed, which helps to maintain food, which is why you have to push the food through the digestive tract. And because it's closed and you have this pushing and you have a hydrostatic skeleton, it puts all of those gut contents under pressure. What does that mean? It means they can shoot their waste some distance if they need to. All right? For Ascarisum, up to 60 centimeters. Who needs an undergraduate research project? See if we can beat it. All right, so that's our gut. That's our digestive system. Yep.
Does anybody know why they're able to shoot that far? Like, what's
It's under pressure.
advantage of that?
Nah, there's probably no advantage.
Okay.
It's just under pressure. And again,
Boost.
yep. And again, I just would like to be in that lab when someone said, let's measure how far it can go. The idea probably formed around a happy hour on a Friday afternoon. All right, osmoregulatory and the excretory system. This is kind of interesting for the nematodes. All right, so how developed that osmoregulatory system is kind of depends on the environment. So if you have free-living forms that especially live out in freshwater environments, they're going to have adaptations that allow them to move water across the cuticle. All right, it allows them to move water across the cuticle, prevent themselves from blowing up from inrush of water. In a lot of our parasites, the adult nematodes tend to be osmoconformers, which means they're putting in whatever their salt and solute concentration is inside the body, that's probably what they're experiencing in the environment, which means they don't necessarily need a very well-developed osmoregulatory system. All right, but they still utilize an excretory system, and a lot of times these excretory systems are paired with the secretory system, where they secrete some compounds. The structure that's somewhat unique, is unique to the nematodes, are these rennet cells. I guess you could probably call them Rene cells as well, or rennet cells. These are ventral gland cells that open to the exterior via a mid-ventral pore. So these are different types of shapes of that excretory system. You've got a single rennet cell, you've got a two-celled rennet cell, two cells. Usually our worms that we see would be these H-shaped pattern where you've got these one or two cells, the rennet cells, that are then have this paired ducts or a paired system that then opens to the exterior. This is unique to the nematodes. And their whole function is to secrete some stuff. Now, what are they actually secreting? Good guess. You can have a good guess. There's been a couple papers that have demonstrated that they're secreting a substance that helps kind of cement one of the worms to an environmental structure. All right, but that's done in a free-living nematode. All right, what is the role in the parasitic stages? Maybe it's tied to host immune evasion. But these rennet cells are unique and are part of the secretory excretory system. And we can't see these on any of our prepared specimens. We can't see them. I should say the location is up at the anterior region of the worm. It's near the nerve ring is where we see them, so it's more anterior cephalic portion. Has a lot of stuff on this slide. So if I tell you we have an unknown and we can identify a rennet cell, and I say what phylum is this parasite most likely in, you should know it's nematodes. Ready? All right, reproductive system. Typically, the nematodes are dioecious, so you're going to have separate sexes. And oftentimes, we're going to see dimorphism in our nematodes. So normally, the female worms are going to be larger than the male worms. What do you think the implication is of that? Or why would the females be larger? For the eggs. To hold more eggs. Yeah, it's, I mean, if females are producing eggs, the bigger you are, normally the more eggs you can produce. That's usually true with most of our dimorphic species. So females are usually larger. Males tend to have a curved tail. And that curvature of the tail is thought that it helps to hold the female during sperm transfer. All right, so what we're going to do is we're going to look at the male system and then look at the female system. So in our male system, usually we have a single testis. And the testis and the ovary in this group are solid and thread-like. All right, so in the Platyhelminthes, you have more of a solid testes in the digenetic trematodes, and then when you get to the tapeworms, they tend to be more follicular, but you can have some solid ones. Like Hymenolopus has these 3 testes inside of it. But instead of being solid and circular or round-like, they're thread-like. So here we have, a single testis starts here, it's thread-like, and then as the sperm get produced, they mature in the seminal vesicle. Our pattern is just basically the same like in any other male system. You have a testes, vas deferens, then the ejaculatory duct leading to the cloaca. All right, so that's our subterminal date. Nematodes can have a variety of accessory structures, but not all of them are present in every species, and you'll see some of this. You'll see some of this diversity. So the one that usually is present is a spicule. A spicule is a chitinized structure whose function is to hold open the vulva during sperm transfer. All right, so the cirrus in the Platyhelminthes delivered the sperm. The spicule is to go in and open up the vulva so that sperm can transfer. Now, the sperm are non-flagellated. Sperm actually look more amoeboid in shape, where they have this kind of, I don't know, what would you call that, flag-like tail. It's called a pseudopodium that will inch them and move them along the spicule. And move them along the spicule. And then move them up the uterus to get to the eggs, to fertilize the eggs. I do say it could be sheathed, so the spicule could be enclosed in a spicule sheath. We're not going to be able to see these. Our scopes aren't good enough, but we will be able to see spicules of a bookworm. Another accessory structure that could be present is called the Gubernaculum. This is a sclerotized portion of the rectum whose function is to guide the spicule as it passes through the cloaca. All right, so it's kind of it's guiding it so that it doesn't start ripping up the back end of the male worm. Guides it through the cloaca. And you could ask, why does it have to hold it open? Again, it kind of goes back to the structure of the nematodes. The cuticles is providing thick structure. We have a lot of internal pressure. So we have this hydrostatic skeleton. That hydrostatic skeleton is partially responsible for holding the vulva closed. So the spicule comes in and holds it open long enough for sperm to actually transfer through. And the gubernaculum allows the worm to actually get that spicule out and potentially into that vulva. Third structure, copulatory bursa. We don't have it. We have hookworms have it, and we'll see the bursa. Its function is to grasp and hold that female. And here bursa, think of almost like an upside down, like an umbrella that is completely wind opened, right? That can now bend and grasp and hold. Not all of them have these. And bursa could be highly developed, could be just almost like those caudal alley, the wing shapes. Ready? All right, so the female system, usually it's didelphic, which means it's paired. All right, and this is why I put the pattern up here. All right, it's paired because you have one vulva, there's one vagina, but then you've got a split, then you have an ovojector, a uterus, an oviduct, and an ovary. All right. So it's paired. You've got 2 ovaries, 2 oviducts, 2 uteri, 2 ovojectors. Just like the testes, the ovary is thread-like, so it extends just basically as a long continuous tube. We don't have an ootype because, we don't have an ootype where fertilization is going to happen, because what ends up happening is usually the junction between the ovary and oviduct, but once we transition, that seems to be where we have our seminal receptacle and where fertilization would actually occur. But you don't have like a Malus gland or anything, encircling that structure. So the uterus is muscular, becomes more muscular at the distal end, and as it approaches the vagina, all right, and that acts as an ovojector. So it retains, helps to hold the eggs inside the uterus, and then it can let the eggs out and kind of expel them. So here is in our diagram, this is didelphic. You've got your vulva that opens up. The vulva would be basically the outer lips. We can see some on some of our slides. If the worm's positioned sideways, you can make out where the vulva is. And then you have your ovojector. You could say thickening of the ovojector. We do have the whipworms where we can see it, and it really is muscular ID because the eggs are single file in there. But in this one, you've got both directions leading all the way to their ovaries. 2 sets of ovaries. Ready? Nervous system. Key thing for this is that we have a circumesophageal nerve ring right at the anterior end. Are we going to be able to see it? Not in any of the specimens that we have. Occasionally in some specimens that we've made temporary mounts of, you can make it out. You can make out this nerve ring. Normally it's located in our esophagus pharyngeal region where it constricts, where it narrows. It's usually where we find our nerve ring. All right. So we have the nerve ring up at the anterior end, and then we have prominent dorsal and ventral nerve cords that run the length of the worm. And then along these nerve cords, you can have these commissures that connect the two. We do have lateral nerve cords that tend to be less prominent. So in our ascaris cross sections, we'll be able to see the nerve cords, the dorsal and the ventral nerve cords. They're very prominent. The nerves connect to sensory structures. So if we have like the cephalic papillae or caudal papillae, those mechanoreceptors will attach to some place on those nerve cords, or to potentially the nerve ring if we are going up to the anterior end. At the caudal end, we have preanal ganglion. So you're talking about potentially for central nerve cells, you can say, that are connected to some of the caudal sensory structures. And we do see a posterior nerve ring, so cephalic and a caudal nerve ring. Are we able to see any of these? Nope, not in any of our specimens. But know that they have nerves. Dorsal and ventral nerve cord run the length. They need to. And what connects to the nerve cords? Sensory structures and what's the second thing that connects to these? The U-shaped muscles. Muscles. All right, we ready for this? Move on. All right, the eggs play an important part of our life cycle. Eggs are going to get out. And in several of the worms' life cycles that we discussed, the eggs are going to be the infective stages. The eggshell itself is well developed and consists of 3 layers. Three main layers, because there could be a fourth. Could be a fourth layer. We have this outer vitelline layer, a chitinous layer, and a lipid layer. Outer vitelline layer, a chitinous layer, and then a lipid layer. The vitelline layer is lipoproteins. That's proteins. The chitinous layer is usually the thickest one. And if we see images of nematode eggs, and you can see like striations, that's our chitinous layer. And then we have an inner lipid layer that really contributes to impermeability. Gives these eggs the resistance. In some species, these eggs are highly resistant to disinfectants, like formaldehyde, bleach, just highly resistant to all of those. And most of those, they have a fourth layer, which is contributed by the uterine cell secretions, which we'll call the uterine layer. This is a proteinaceous layer. All right, proteinaceous layer made up of acid mucopolysaccharides. So you've got a layer that blocks out some of the fats, some of the polar compounds, but then immediately underneath it, you've got lipoproteins and a lipid layer that blocks out anything that could get through this protein layer. Makes them very, very highly resistant. Heat will kill them. Freezing doesn't always work. Bleach and formaldehyde, not so much. Ascaris eggs. Hopefully all of you that dissected an ascarid worm in zoology, I don't know, maybe you didn't get to, because maybe that was online that semester. But hopefully you washed your hands before you started eating after that. Because any of those females, if they were shedding eggs, those could have been infective. Highly resistant to chemical disinfection. We typically don't see this much development in eggs. All right. We're good? So that's leading to our life cycle. Generalized life cycle, very, very easy. We have our egg. Egg gives rise to 4 different juvenile stages, abbreviated J1, J2, J3, J4. All right, so of our juvenile stages, which one will be more developed, a J3 or a J2?
J3.
Yeah, J3. Again, just goes in order. We're increasing our development. All right, and then the adult stage. Do you remember this and keep these down? We're good. All right, and this applies to all of the nematodes. So our growth between the different stages is accompanied by cell enlargement and ecdysis, which is our molting of the cuticle. So our egg hatches, releasing the J1. In order to transition to the J2, we have to molt. In order to go from J2 to J3, we molt. To go from a J3 to a J4, we have to molt. And then from a J4 to the adult, we have to molt. All right, so there's going to be 4 different molts happening in this parasite, or in these life cycles. In some cases, when the worms molt, they retain their cuticle. So if you have a J2 that molts to a J3 stage, they may retain that J2 cuticle. What would be the benefit of that? Increases the level of protection. All right, so we'll talk about that. It's kind of an adaptation for transmission. In a lot of our life cycles, in a lot of nematode life cycles, the J3 is usually the infective stage. So if you're unsure of, well, what stage is infective to the definitive host, if you answer J3, oftentimes you're going to be right. I would put it in a catch because the parasites that we talk about, that isn't always the case. So, but I just wanted to point out that usually the J3 is our infective stage. So that's our generalized life cycle. But we do have a lot of diversity. Not so much in the general pattern, but we have diversity in what hosts are infected, do we even need hosts, how do we get from one host to the next, and so forth. All right, so in our reproductive strategies, we're going to talk about direct and indirect life cycles. We're going to have both. We're going to see both facultative and obligate parasitism in the nematodes. Some of our nematodes require vector transmission, which is we're relying on an insect host to get us from one host to another. So our insect is serving as a vector for transmission. We could see diacy, parthenogenesis, haplodiploidy in nematode life cycles. And we have some life cycles where we will see tissue migration. So you get to the host, you're in the gut, and then it moves out of the gut and undergoes this circuitous migration only to get back to the gut, where that is the final site of infection. So there's a lot of diversity to the life cycles, even though the general pattern stays the same. Even though the general pattern stays the same. Questions? All right. So next up that we're going to do is introduce diversity of our life cycles. And the first thing we're going to talk about are the nematodes that infect invertebrate hosts. All right, we have a couple life cycles to review. These are important nematodes because they usually serve as regulators of insect populations. Some of them have been investigated as biological control agents of these insects. And then what we'll do is we'll split and start talking about vertebrate hosts. Now with the vertebrate hosts, we have basically one of two types. Those that are stichosomes bearing, which are like a lot of glandular cells. Those that aren't, like just your regular Ascaris type nematodes and hookworm type nematodes. And then what we're going to wrap it up with are those that require a vector. Because now we've undergone evolution of life cycles to basically pick up another host that allows us to transmit. All right. So, any questions? And I apologize because I posted this next handout, but I didn't make copies because I honestly didn't think I'd get up to this point. We've got 10 minutes. I hate to end it. I think what we'll do is we'll just introduce it, the invertebrate host, and that's it. We're not gonna, there's some life cycles that we'll address, we'll get it on Wednesday. So, as I said, we're gonna deal with invertebrate definitive hosts. And it has arisen. Utilizing inverts as the definitive host arose at least 4 separate times, and they arose independently. But these all arose first, and then there was a transition to vertebrate hosts. So all of our invertebrate host life cycles, they're basal to our vertebrate host life cycles. There's three main families that primarily infect insects. There's the Steiner Nematodae, the Heterorhabdidae, and the Myrmithidae. All right, we're going to talk about two of them, the Steiner Nematidae and the Myrmithidaes. Members of Steiner Nematidae and Heterorhabdidae exhibit similar life cycles, where they utilize bacteria as an endosymbiont. They actually rely on bacteria for their life cycle. We're going to talk about these because of their potential to control insect pests. We do not have examples of these. But if you were to take like entomology, there's a potential that you would find some of these parasites if you looked at them. But I'm sure you didn't look at them because you didn't have the best class here at ASU yet. That's what you're learning about right now. All right. So we're going to stop here. I just want to give you the introduction. What we're going to do is talk about Steiner Nema, and then give you two other of these life cycles. So there's going to be three life cycles that we cover. In the Myrmithidae, there's an aquatic life cycles and there's terrestrial life cycle patterns. So we'll see them. We'll talk about them. All right. I'm going to go down and make sure the lab is open if you want to do some last minute studying for the lab practical. Check your time, 2 o'clock, 3 o'clock. I'll have the door closed, so just hang out in the hallway, and then what I'll do is I'll let you in. We're taking the practical. That's what we're doing. Once you're done, you're done. All right.